The realm of genetic engineering paves the way for the modification of both animal and plant genomes. This is achieved through a series of strategic techniques that empower scientists with the ability to alter DNA at various levels. From a specific gene to an entire organism, creating a genetically modified organism, GMO, demands a series of steps, commencing with the selection of the gene for mutation. This chosen gene can then be isolated and assimilated into a fitting vector, an essential component that enables the gene's integration into the host genome, thereby birthing a transgenic or revised organism. Unraveling the mysteries of gene function and manipulation have been the cornerstones of crafting the science of genetic engineering. Notable milestones include the discovery of restriction enzymes, DNA ligases, the development of sequencing, and the polymerase chain reaction. Genes introduced in this form often bring along promoter and terminator regions, with potential modifications to enhance efficiency. The newly introduced vector then integrates with the host organism's genome. In animals, this gene insertion typically happens in embryonic stem cells, whereas in plants, any tissue that can develop into a full-grown plant is fair game. Both instances subsequently undergo examinations to assure stable gene integration, inheritance, and expression. The first generation of offspring would be heterozygous, necessitating inbreeding to yield the homozygous pattern corresponding to stable inheritance. Initial methods projected genes into the genome randomly. However, advancements allowed for targeting specific locations, mitigating unexpected side effects. Since 2009, systems engineered for precision witnessed an uptick. Among the most common are transcription activator, like effector nucleases, TALENs, and the Cas9-Giderna system perfected from CRISPR. The evolution of genetic engineering is propped up by numerous discoveries and advancements. The genesis of man. Managed genetic manipulation dates back to 12,000 BC, catalyzed by artificial selection in the domestication of plants and animals. Techniques such as hybridization orchestrated swift changes in the organism's genetic structure, a tool which was most commonly used when humans commenced planting genetically diverse individuals of related species in adjacent proximity. One of the key landmarks in understanding genetic inheritance can be traced back to 1865, owing to Gregor Mendel's P-crossing experiments. In 1928, Frederick Griffith unearthed the existence of a transforming principle complicit in inheritance, with DNA being identified as the said principle in 1944 by scientists Oswald Avery, Colin MacLeod, and McLean McCarty. Finally, Frederick Sanger, navigating the course of DNA sequencing in 1977, revolutionized genetic engineering by empowering researchers with a significant volume of genetic information. The story revolves around developing techniques to integrate DNA into an organism's genetic structure alongside its origination. Griffith's experiment had revealed bacteria's capacity to absorb and utilize alien DNA. Researchers induced artificial competence in Escherichia coli in 1970 through calcium chloride solution. The late 1980s saw the advancement of transformation using electroporation. Researchers in the 1970s observed that the bacteria Agrobacterium tumefaciens, discovered in 1907, could integrate its DNA into plants. This integration was possible through a Thai plasmid. By substituting the tumor, causing genes in the plasmid with new genes, researchers managed to infect plants with a tumefaciens. This allowed the bacteria to introduce their selected DNA into the plant's genomes. Identification of the target gene was the first step, hinging upon the eventual objective of the derived organism depending on the complexity of the goal. Even entire biosynthetic passages involving numerous genes might be altered. Upon identification, genes and genetic data from diverse organisms could be inserted into bacteria for safekeeping and alteration, creating genetically modified bacteria. These bacteria, inexpensive, easily grown, clonal, and capable of multiplying rapidly, could be transformed and stored at very low temperatures. These modified bacteria harbored the separated gene, providing a limitless supply for investigation. Using genetic screens and tests, scientists could discern the potential genes and identify the most suitable candidates. A rather straightforward genetic screen would randomly mutate DNA using chemicals or radiation before selecting the ones exhibiting the desired trait. If mutation wasn't viable, researchers would search for naturally occurring mutations within the population. These twin processes involving the examination of a phenotype and identifying the related gene, termed forward genetics, required careful mapping of the gene by tracking the inheritance of the phenotype against known genetic markers. Genes located close together are more likely to be inherited together. An alternative approach, reverse genetics, involved targeting a certain gene with mutation, then observing the resulting phenotype. The mutation could be designed such that it deactivates the gene or activated it only under certain conditions. This was particularly useful for identifying genes that are fatal if non-functional. Primarily, genetic engineering in animals utilizes growth hormone genes. This intricate process involves the alteration of DNA traditionally sourced from cellular organisms. The creation of a DNA library later enabled genes to be cloned from a DNA segment or artificially synthesized for the gene to express itself in the host organism. Additional genetic elements are added, which also facilitate selection. Firstly, the cell is delicately opened to prevent significant damage to its DNA. This methodology fluctuates based on the cell type. Upon opening, the DNA needs to be isolated from other cellular elements. By using a blend of phenol and R-chloroform, followed by centrifuging, nucleic acids can be separated into an upper aqueous phase, devoid of any cell debris. Further purification can be achieved by repeating the phenol-chloroform steps, and finally the nucleic acids are precipitated using ethanol or isopropanol if necessary. RNA can be eliminated by adding a ribonuclease. Currently, companies are simplifying this complex process by selling specific kits. The gene that scientists aim to modify, the gene of interest, has to be separated from the extracted DNA. If the sequence is not known, the common practice followed is to divide the DNA using a random digestion process involving the usage of restriction enzymes. These enzymes cut the DNA into overlapping fragment segments, which are then placed individually into plasmid vectors and reproduced inside. The bacteria, if the desired phenotype, can be identified within the DNA library. It could suggest that the target gene is present in the bacteria in case the gene does not exhibit a recognizable phenotype or the correct gene is absent in the DNA library. Alternative methods are applied to isolate it. This could include chromosome walking if the location of the gene is known, or looking for closely matching genes in the library if there is high homology to an identifiable gene from another species. For a known DNA sequence, restriction enzymes that make incisions on either side of the gene are used, following which Gel electrophoresis orders these fragments. By length, the DNA can be visualized by staining with ethidium bromide and photographed under UV light. It's important to note that while the polymerase chain reaction efficiently amplify a given sequence, its effectiveness decreases with larger genes, and it might also introduce sequence errors. Artificial synthesis of genes has further simplified the process, with several synthetic sequences being commercially available. The primary focus of this narrative is the process of gene insertion into a host organism's DNA, which can occur either as integrated genomic DNA or extrachromosomal DNA. Various methods for gene insertion are suitable for different types of organisms. For instance, in multicellular eukaryotes, when the introduced gene is integrated into the host germline cells, these cells can consequently pass the changed gene to their offspring. However, when the transgene is integrated into somatic cells, it cannot be inherited. Transformation, a process which involves the direct manipulation of a cell's genetic components through the transfer of genetic material, becomes necessary. Roughly approximately 1% of bacteria naturally possess the ability to absorb external DNA. However, it's possible to prompt this capability in other bacteria through the application of heat shock or electroporation. This makes the cell membrane susceptible to DNA, which may be incorporated into the genome or exist as extrachromosomal DNA. Electroporation, a method that exposes cells to an electric field, also assists in making cells competent. In this process, plasmid DNA can enter the cell through the holes created in the cell membrane during the electric shock. Once the DNA has been taken up, it can either fuse with the bacterial genome or remain separate, typically as extrachromosomal DNA. For plants, the common method of DNA introduction is agrobacterium-mediated recombination. This involves the use of the agrobacterium's tDNA sequence, which facilitates the natural insertion of genetic material into plant cells. By altering the plasmid to express the gene of interest, researchers can stably embed their selected gene into the plant's genome. This entire process forms the main driving narrative of this story. The process of giving animals extraneous DNA, known as transfection, often signifies a shift towards cancerous development. Diverse methods exist for the direct injection of DNA into animal cells in vitro, most frequently using stem cells intended for gene therapy. Essential to this procedure are chemical-based techniques employing either natural or synthetic compounds, creating particles that aid gene insertion into cells. These synthetic vectors are capable of binding DNA and facilitating large genetic transfers. A straightforward method uses calcium phosphate to bind the DNA before exposure to cultured cells. The solution and DNA within become enclosed by the cell's aids for transporting DNA into cultured animal cells include liposomes and polymers. Liposomes with a positive charge bind with DNA, whereas polymers can be crafted to interact with DNA, forming lipoplexes and polyplexes respectively. These are then absorbed by cells. Alternatives include electroporation and biolistics. In certain instances, transfected cells may stably incorporate external DNA into their own genome, a process known as stable transfection. Creating transgenic animals necessitates DNA insertion into viable embryos or eggs. This is ordinarily achieved through microinjection, where DNA is administered directly into the nucleus through the cell's nuclear envelope. Superovulated fertilized eggs acquired at the single-cell stage are then cultured in vitro. Once the protoplasm reveals the sperm heads per nuclei in egg, the genetic material is introduced into one of them. The oocyte is then planted in a pseudo-pregnant animal's oviduct. Alternatively, embryonic stem cell-mediated gene transfer can be utilized. The gene is transfected into embryonic stem cells before implantation in mouse blastocysts, which are subsequently placed in foster mothers. As a result, the offspring is chimeric, and additional mating can yield mice that are fully transgenic for the desired gene. Transduction represents the procedure by which a cell is given foreign DNA via a virus or viral vector. Genetically modified viruses can serve as viral vectors, transferring target genes to another organism as a form of gene therapy. Initially, the virus's virulent genes are excised, with target genes implanted in their place. The sequences facilitating the virus's gene insertion into the host organism must remain undamaged. Common vectors derived from viruses include retroviruses or adenoviruses, while other viral vectors sourced include lentiviruses, poxviruses, and herpes viruses. The virus used depends on the targeted cells and the permanence required for the DNA alteration. In the intricate world of genetic engineering, specialized cells carrying DNA markers provide a valuable tool for identifying the presence of certain genes. When these cells are grown in a specific antibiotic or chemical environment, it enables us to distinguish genetically modified cells from their unaltered counterparts. Alternatively, a DNA probe designed to bind only to the inserted gene can be employed for the same purpose. Often, these markers linger in the transformed organism, though we've devised strategies to remove them from fully developed, engineered plants. But the journey doesn't end with simply ascertaining the existence of inserted genes in a given organism. To ensure they're activated appropriately in the targeted tissues, further probing via PR, southern hybridization, and DNA sequencing is undertaken. These tests validate the new gene's position within the chromosome and its number of copies. Additionally, an array of methods, including northern hybridization, quantitative RT-HETR, Western blot, immunofluorescence, ELISA, and phenotypic analysis, are engaged to evaluate gene expression and related aspects. The subsequent generation of the organism is also examined for the stable inheritance of the transgene and its corresponding phenotype. Traditional genetic engineering methods, which involve arbitrary insertion of new genetic material, can potentially disrupt or modify the host organism's genes. To overcome this, techniques were invented allowing for pinpoint insertion of new genetic material into desired locations within an organism's genome, a technique primarily leveraging the principle of homologous recombination, HR. This approach, specifically applied to embryonic stem cells, facilitated the production of transgenic mice with targeted gene knockouts. Remarkably, this has made it possible to either introduce genes or tweak gene expression patterns. In situations where suppressing an essential gene could be fatal to the organism, alternative methods such as site-specific recombinases, SR, became necessary. The most widely used SRs include CRI, LOXP, and FELV-FRT systems, which function by removing DNA via an enzyme-mediated homologous recombination process. By pairing an organism containing recombinase sites surrounding a gene of interest with one that expresses SR under the control of tissue, specific promoters genetic modification becomes a controlled process. This methodology has been instrumental in deleting marker genes from transgenic animals and even enabling conditional recombination to switch on or off genes at specific times or developmental stages. Lastly, genome editing employs artificially engineered nucleuses to induce precise double-stranded breaks at desired genetic locations, thereby augmenting our precision and control over genetic engineering experiment. The research has been increasingly focusing on devising tactics for creating gene knockouts or corrections without the need to create double-stranded breaks. Meganucleuses, first used in 1988 in mammalian cells, are restriction enzymes with long recognition sites, contributing to their specificity likewise. Zinc finger nucleuses, introduced in 1996, possess ends. Introduced in 1996, possess enhanced specificity courtesy of the fusion of zinc finger domains and Fuki nucleus. Domain. The subsequent highlight of the research was the discovery, in 2009, of a new group of efficient gene editing tools involving the code governing DNA recognition by tail. This is a protein secreted by the Xanthomonas plant pathogen and binds with specificity to genes. Engineering tail resulted in the creation of tail nucleus, TALEN, which is so far considered to have one of the highest specificities amongst engineered nucleuses. The latest major development in this field, in 2011, further amplified the efficiency of gene editing, which saw the emergence of CRISPR system. This system, functioning as an adaptive immune system, helps bacteria and archaea to protect themselves against viruses by cleaving viral DNA. Over time, this mechanism has been modified and utilized to induce double-stranded breaks at specific points in DNA sequences, leading to insertion-deletion type mutations in most instances. In the rapidly evolving world of gene editing, base editing techniques are being honed. These involve the use of AI nucleus, DED's Cas9 endonuclease, or a similar enzyme for targeted gene manipulation, with an attached deaminase enzyme instigating specific changes in the DNA base. A cutting-edge advancement in this field is the innovatively designed method called prime editing. The approach attaches a reverse transcriptase to an RNA-guided specially engineered nucleus that makes single-strand cuts, but abstains from double-strand breaks. The segment of DNA adjacent to the cut is replaced through the sequential activity of the nucleus and the reverse transcriptase, which executes the desired alteration using an RNA template.